Hi, I'm Andrew Nenka. Thanks for coming to listen to this talk. I'm going to be talking to you about some of the infrastructure we've built in the Center for Imaging Research, specifically about building a segmentation deep neural network to work on your own data. We're going to talk through just an application to some of the projects that have happened in my lab, but I'd really like to give you an overview of the tools that we have and that are available for researchers in the Center for Imaging Research here at MCW to use. So before we actually jump into the infrastructure of the Center for Imaging Research and the applications in my lab, I just want to take a couple minutes here to talk about the ubiquity of deep learning, machine learning, and its infiltration to the medical practice of radiology. So first off, why is it so ubiquitous? And it's really just the fact that this deep learning technology has made things that were previously unthinkable reality. So a perfect example is autonomous cars, right? This image on the right is showing a view of a street from a car with the segmentation that can happen in real time of not only where the street is, but where are the pedestrians, where are the bicycles, where are the other vehicles, all of which, of course, is necessary for self-driving vehicles, all of which as well, a decade and a half ago, to see something like this happening and happening in real time would have been insane. Part of what has made this possible has been great advances in computational hardware. The general purpose use of graphical processing units allows for the tremendous number of multiplications and additions that happen in the training of deep neural networks and the application of deep learning neural networks to actually perform those calculations at timescales that are reasonable for research and for applications. Of course, hardware is nothing without the software to go with it. And over the past many years, Google has come out with their TensorFlow API and made it open source. So all the deep learning technology that sits behind Google is available for researchers and pretty much anybody in the universe to use. The same thing has happened with the technology that's been developed at Facebook with PyTorch, and in fact, a large number of other deep learning APIs or application programming interfaces have been created and opened up to the full community to allow for further development and further use of those tools. With the tools being open source and available, then the derivatives have also tended to become open source and available. So as new networks have been developed, including the segmentation nets like the VNet or the UNet, which currently leads the field, people have been writing this code using the open source tools and then releasing their software to other developers to continue developing, improving, and distributing for others to continue using. Of course, nobody likes free stuff that works really well more than an academic. And our world has really embraced this high-performing open source software. If you just hop on to Google Scholar and search for deep learning, we have like 4.8 million results, and this field is continuing to grow exponentially, driven very much so by the hardware that allows this work to happen, the software that allows people to write good code that uses that hardware and perform the convolutional neural networks that are used in machine vision, and the releases of software that are built upon that all that gets down to something that becomes very useful for end users. So with all those advances in deep learning in the general sense, why has it really taken off in medical imaging? And the answer simply becomes one that deep learning excels at it. Machine vision is sort of the main use case that people point to with deep learning these days. And many have come so far as to say that deep learning has solved the problems of machine vision. What wasn't possible previously now is and is quite generalizable. Now, machine vision can answer many of the tasks that are performed in radiology. And I'll just take a second here to talk through some FDA cleared products using deep learning that exist for medical imaging. So there's the problem of classification, looking at an image and deciding what's there in it. And one of the first things to come to market has been this idea of reviewing with a computer all head CTs that come through and identifying those who have a intracranial hemorrhage. And if the machine learning identifies that there's a hemorrhage in that patient, not necessarily making a diagnosis of it, but taking that exam and putting it to the top of the work list for the radiologist, so that when there's an acute finding that has actionable information that can really benefit from those images being looked at early, to allow them to be there for the radiologist to look at sooner rather than later. Now, there's been also this idea of radiomics, looking at the texture of images, and in fact, that becomes or has been proven to be something that's useful in mammography, looking at the breast tissue density, looking with those films from the mammograms, applying radiomics to classify the density. And segmentation, right? Segmentation is taking that image and breaking it up into regions of interest. One case, of course, is with pneumothorax. If you have air in the thorax, you have problems. And how much air is there can be a useful measure, something that a radiologist would often have to do if they're presented with a CT that shows it. But you can create a segmentation using deep learning that identifies that region and measures it for you with a precision that really outpaces that which a radiologist can do in a quick amount of time. And with that segmentation and volumetrics, of course, in this group, it's probably not going to be a hard cry to talk about brain segmentation and looking at volume of different regions of the brain, correlating that with mild cognitive impairment and general dementia cases. There, again, deep learning can really segment that brain, give quantitative values for regions of the brain, which can be used to identify different, or in subclass, different types of dementia. And then, of course, the stuff that I do and that the other physicists around me do is the crazy under-the-hood math stuff for image generation. And GE's gotten recent FDA clearance for a deep learning image reconstruction for MR, allowing one to get higher resolution images with less noise and less time on the scanner. So really, in fact, doing something that those who are watching the books care about, increasing throughput, but in doing it in such a way that you actually maintain data quality, image quality for the radiologists reviewing the exams. So with all those applications already out there and driven to market, one gets to the point of looking at deep learning in medical imaging and asks themselves the question, in my research work, do I want to be doing work with deep learning or do I want to be doing research work on deep learning? And there's a mixture of answers. In my lab, I can say I answer both yes. But just in so far as an example, right? If you look at the scientific reports article, it was recently published describing a deep convolutional neural network to segment white matter hyperintensities, and talks about the development of that algorithm and trying to find, create a better mousetrap for finding those white matter hyperintensities, segmenting and doing quantitative analysis on them. Conversely, this AGES study has gone so far as to take the trained neural networks that exist for white matter hyperintensity segmentation and just apply them, use them as a tool so that in a study of elderly populations, they can get those volumes of the white matter hyperintensities without having to manually segment them, but still do it in a way that is accepted and that is quite reproducible thanks to the use of an algorithm for the segmentation. So with this technology now of machine learning being available not only as something to do research on, but to do research with, nearly 13% of all of the ISMRM abstracts that were just submitted or presented in the 2021 conference included machine learning as a component. Taking a further step over to the world of more applications, in the Radiological Society of North America meeting in 2020, over a quarter of all of the educational exhibits included machine learning. So this technology exists. It's not just something that people are doing development on, but it's matured to the point where, in fact, it's permeating most of the or much of the research work that's happening. And here at MCW, it's not really any different from the rest of the world. So I just have a few examples of things here, and my apologies to not being able to offer an exhaustive list. But the fact is that so many people are doing work with machine learning here that I'm invariably going to end up missing folks. So the folks work in Yang Wang's lab is doing fMRI prediction based on machine learning. Work happening in Dr. Pete LaViolette's lab is doing segmentation and characterization of various cancers with deep learning and aligning those imaging results with pathology results. Also, his lab has done some interesting work in the techniques of machine learning, identifying ways to improve the training process. Kevin Koch has been working in his lab to use deep learning to solve that crazy difficult inverse problem of quantitative susceptibility mapping with his former graduate student now, Juan Liu, developing all sorts of neat neural networks to do that regression problem. In my lab, I've been using the deep learning to perform reconstruction, similar to the models of GRAPPA. In fact, replacing the GRAPPA convolution with a convolutional neural network and doing some, again, fun things with training to improve it. And then in this last image over here on the right, just to show the diversity of what's happening here, if you look for researchers from MCW that are publishing their preprints with deep learning on arXiv, there are at least 8 there right now showing different applications and technology development with deep learning coming from imaging folks here at MCW. So following all those examples, I'm going to really narrow down the rest of the talk here to be focusing on research with machine learning. And really, one of the questions becomes why use machine learning in your research? And really, it's a tool to avoid a lot of tedious pre-processing of data that in some cases becomes prohibitive with the resources that we have. So if you're doing a region of interest based analysis, you need to define those regions of interest, of course. While the brain has templates, elsewhere in the body does not always have templates. So the question becomes, how do you define those regions of interest? And one thing that you can do is train up a neural network to draw those regions of interest for you. Those regions of interest, of course, if the training data is very good and matches up with your experimental data, you can create some really high precision regions of interest, and you can do a whole bunch of them. The clock cycles on a computer are cheap and easy to run in comparison to somebody drawing those regions of interest by themselves. Beyond that, using an algorithm to define those regions of interest makes them reproducible and consistent, something that really helps drive the field forward, having that reproducible tool. Also, if you have a professional draw those regions of interest, right? A highly trained radiologist can read that image and identify things in there with far higher precision than many of us mere mortals. To have them curate that data set allows the machine learning to pick up their skills and start drawing regions of interest with the precision that they tend to use, or create regions of interest that they can then go back and just tune up rather than having to draw manually by themselves for the full data set. And that's really important when those professionals are, of course, in short supply with extreme clinical requirements. But altogether, really you want to hop into this machine learning as a tool for your research is if the standard techniques of machine vision are out there and are too complicated or just not sufficiently sophisticated to be practical or to be applicable to the data sets that you have. I've seen my first example that I'll talk about in a few slides here is an example of killing an ant with a heat guided missile. Insofar as the standard techniques would have certainly worked in place of the machine vision techniques or in the machine learning techniques, but we went down that road for other reasons. But really, sometimes a quick and simple solution can be put forth with traditional machine vision techniques that can actually streamline your process a lot more than training up a deep neural network. But when things start getting complicated, those deep neural networks are outstanding. Of course, you can use machine learning for complicated regressions, nonlinear regressions. The whole quantitative susceptibility mapping thing that's happening in Kevin's lab and the GRAPPA or the Rocky neural networks that are happening in my lab are examples of that, but we're not going to really jump into that. And then, of course, there's the whole coolness factor, right? I mean, if you look at how many things are getting published with deep learning and machine learning, obviously there seems to be a bias to seeing things like that accepted and published. So including the latest and greatest is always a nice feather in your hat for your research work. So now that I've theoretically convinced you that you should be using machine learning in your research as a tool more so than as a topic of research, I guess the question becomes, what are the right steps to do it? And first off, of course, you need to define the science, right? Rather than creating a hammer and looking for all of the nails, take a look to see if machine learning really makes sense with the scientific question that you're looking to solve. And once it does, because obviously it will, identify the sources of data that you're going to be using for your project. Right? Are you going to be using open data that's available on the internet? Are you going to be pulling clinical data in perhaps from PACS or from someplace else? Are you going to be using research data sets that already exist in your lab? Depending upon where that data is coming from, there's certainly going to be different pre-processing steps that are needed to pull that data into a format that'll be conducive for the machine learning training. Then it's always an important thing to consider where you're going to be doing this work, and if those images coming in have protected health information, and if that health information needs to be managed in such a way that makes it appropriate for doing your work on it. Once you have that data identified, of course, you need to organize it, annotate it, and then develop the model, train the model, test the model, find out that it doesn't work, and try again, and try again, and try again. And once you get to that point where it is working, apply it in production. And then at the end, of course, publish that work and continuing with the open model of the field, share that model with other investigators. And now we finally get to the punchline, or the whole purpose as to what motivated me to talk through this particular work in this forum with the Center for Imaging Research, to talk through the infrastructure that we've built using a couple of use cases that we've had here in the center to really build a generalizable framework that can be used by researchers throughout the organization to streamline this process of training out these segmentation models that we're using. So we've put in place, and we're going to talk about infrastructure for that de-identification of images, the organization of the data sets, the annotation of the data sets, working to train those models, test those models, perform the iteration, and move into production. So before we had any of the infrastructure built in the CIR, I started a project in my lab just to get familiar with this idea of using deep learning as part of a research project. And so we created for the concussion studies here a little deep neural network to find eyeballs in T2 weighted images. The underlying science was that we had a hypothesis that there was a gaze instability in participants with concussion, and that gaze instability could show up as artifacts from the motion of the eyes during those long T2 weighted acquisitions. So the question became, are artifacts around the eyes in concussed participants greater than those in healthy participants? The answer ended up being yes, but we didn't really move forward that much beyond an abstract because nobody really seemed too excited about the results. However, it's a good example of the process that I described on the previous slides that we can talk through. So the question becomes, right, does the science and machine learning make sense? And nobody really wanted to draw 400 regions of interest around eyes and 400 control locations. So yeah, that seemed like a good thing, and that we'd end up with some efficiency here, using a deep neural network to actually identify those regions of interest rather than having people sit and draw them. For the source of data, it was research data set, which we already had in XNAT, where it was already coded and already organized with the project. To do the annotation, because we didn't have any real good way of doing it, I actually wrote a point and click little GUI in Python to drop regions of interest around those eyes and actually some other anatomy of interest to use as a control. But there we were on our own to write that software in such a way that it could create the data for use in the training. Then we identified and trained a model. We wrote a version of AlexNet, which is one of the first deep learning networks for image identification using TensorFlow. It's a non-trivial process to write that model, and in fact AlexNet is pretty basic. And the more advanced, higher performing networks that are out there will perform better, but we didn't have the expertise to write them. Beyond that, we ended up training the model on a $2,000 graphical processing unit in one of our own workstations, which was used for the first time really for the general purpose computing when we did this processing. We tested the model, applied it on a data set, and manually checked it, again, writing our own software to take a look and see how those results ended up coming through. And then we manually retagged cases that added that were wrong and added them back to the training set, which is still continuing to be a pretty nice way of tuning up models for particular data sets. To apply the model, we did it. We did we computed those regions of interest and identified statistics that describe the eye motion, made a graph and everything, and of course, published the work. Interestingly, here I do have these various tools that we've created available in GitHub. And those are private repositories, but of course, if somebody wants access to it, if they let me know, I can certainly share. Just to point a little towards the science, more so than the underlying engineering and infrastructure that most of the talk's about. It worked. We were able to identify eyes. Those red things are covering the orbits and were identified by the deep neural network. For controls, we had the inner ear structures, which were readily affixed to the skull, so that we'd be able to see general head motion differentiated from eye motion. So we had the deep neural network also find those, which are shown on the right image there. And we looked at 4 different time points following the concussion and controls and injured athletes, looking at the inner ear structures in those green areas, not really seeing much change time point to time point. And looking at the eyes, the eyes altogether showed a little more motion artifact than the inner ear structures. And for the concussed athletes at that first time point within 24 hours of injury, we actually saw a significant increase in that artifact that was associated with motion. When it was all said and done, though, we saw that there were certainly opportunities for improvement. Things worked very well with a well-curated research data set, but there are a limited number of sizable research data sets for doing this deep learning. The infrastructure needed for scaling up with clinical data sets would be pretty significant and was not available at the time. Also, because we rolled our own annotation tools, we saw that using something that's robust and user-friendly for that annotation would be a huge benefit, because it would lower the barrier for entry for annotation. Let folks who are not as familiar with digging into the code to actually do that painstaking process of annotating those images. Beyond that, we saw that we're end users of this technology. We're not at the leading edge of writing the own networks. So using other people's networks would certainly be a good thing. Beyond that, it seems a little excessive to buy a $2,000 graphical processing unit and use it once every few years when you have a task that could use it. So developing the infrastructure in a way to use other people's GPUs or communal GPUs would be a good thing. And finally, robust and user-friendly validation tools would certainly be helpful. So the second use case that we developed for deep learning segmentation was really built in a way to address the concerns that we'd seen as we tried to do the manual work and build a pipeline that would be applicable to other projects here at MCW. So while I'm going to be talking about cross-field and cross-subject instrumented hip registration, the work that we did for segmenting those hip implants out is really quite general and really built to be user-friendly and available for other folks in the center. So the hypothesis for the science was that different implants of different compositions yield different artifacts at 3T versus 1.5T, and that some hip implants might make sense to image at 3T and some certainly do not. But to do this, we needed to register those hips in patients that were imaged at 3T and 1.5T and across the various patients that were imaged at both field strengths to get maps of or comparative maps of those artifacts that come off of the implants. But registration, of course, is non-trivial. Good first guess is important. So there we came to the realization that we first need to just more or less align the implants and then worry about aligning the tissue. Of course, that's also further complicated because in comparison to heads, which a lot of people register, hips are articulated. So the femoral component and the acetabular component need to be considered individually. Now, because we thought that we needed to first align those implants, we needed to segment the implants out. And that segmentation is non-trivial. We first tried a lot of standard machine vision stuff with the simple Insight toolkit. And found that it really is a non-trivial process. So we then stepped up to training a neural network to do the segmentation. So going through that deep learning checklist, if you will, for this use case, right? Defining the science. Does machine learning make sense? Yes. In this case, the traditional machine vision wasn't working out, and that process of manually creating ROIs around those implants is quite tedious and taking a great deal of time for each individual implant. Then we identified the source of the data. So we had both research 3T imaging and the coupled clinical imaging data sets that needed to be extracted from PACS for the 1.5T imaging exams for these particular patients. And because those were coming from the clinical record, we needed to take patient privacy seriously and de-identify those images and code them in such a way that they lined up with the 3T images for each patient. So that then led to the organization of the data set, linking those de-identified clinical images with the research images in that XNAT database, annotating the data set, so building that training set where a medical student could draw regions of interest around those implants in a relatively user-friendly interface. And then training the model. Many versions of the leading VNets exist, including, thankfully, an open source version from NVIDIA that we ended up using. And then we learned to deploy that model using containers to allow that to be run universally across any system that has an NVIDIA GPU. We tested the model. We used some easy visualization with Jupyter Notebooks. We had the pipeline available to iterate to manually resegment failed cases and again add them to the training set as that human in the loop training process. And then we applied the model. We did that segmentation and then went ahead and did the actual science, right? That registration and comparison of signal dropout across implant type and field strength, where we did some statistical tests even, which is always fun. And then, of course, we've put this out as an ISMRM abstract. And again, the underlying code is up in my lab's GitHub repository, which I can gladly share. So on to the infrastructure for this work. So first of all, right, this is in the Department of Radiology. We've built a technique to get that clinical data extracted from the medical record. Altogether, the PACS infrastructure is great for viewing images. I mean, that's what our radiologists do all day, but it's not great for extracting images. Now, there are IRB implications here for pulling things out of the medical record, and it's complicated but manageable. In radiology, we've built a couple of very nice, robust protocols with our research coordinators to do this sort of work. But once it's actually through the regulatory approval, being able to script the pulls is important. So our programmers, Brad and Robin, have created DCMTK scripts to actually create a DICOM node on an iMac that can query PACS for session numbers and then pull those exams to the iMac and extract those data sets. And again, this is, although I'm talking about things in the CIR, this particular workflow exists primarily in radiology, driven by the research coordinators and the programmers that are in radiology here at MCW. So once images are pulled from the clinical record, the next step is to make sure that they're safe for our research use without putting those research participants or patients into risk, and that involves de-identifying the images. So the nice thing about standards is that they're easy to follow, and the DICOM format is not just an imaging format, but it's a full set of standards for transfer and de-identification of images. So thankfully with that, our programmers here have been able to create a DICOM node that sits in the Center for Imaging Research. And this DICOM node receives images from other DICOM nodes, including that iMac that pulls data from PACS. And based upon the project to which the DICOMs are sent on that DICOM node in the CIR, applies some defined metadata modifications, and that includes DICOM compliant de-identification of the tags that are in those DICOM images if needed. Once those modifications happen, the node then sends those images on to another DICOM node, which ends up being XNAT for much of the research use. In fact, if you use any of the human systems in the CIR, you're already using this DICOM router. The images go from the scanner over to this router, and this router then sends the images to the right XNAT for your project. So once the images are in a usable format, the next step is to organize them, and we can do that through XNAT in the research enterprise here, which we've already talked about a little bit. Now, the XNAT installations that we have here at MCW can be a little confusing at times. So I just have a quick overview of what we have. We have two major branches, right? We have the branches of XNAT that are available on campus, essentially behind our firewall. And there we try to balance the studies across the two servers that we have, SIR-XNAT1 and SIR-XNAT2. These systems utilize the disks that are in the MCW Research Computing Center for storage. And in that way, we have it built so that your data for your imaging studies actually end up in your group directory on the Research Computing Center's hardware. With the data over here, it becomes very easy to use this data with the high-performance computing hardware that's in the Research Computing Center. Now, for off-campus, we're a little more restricted, and we put things on dedicated servers, GWXNAT and GWXNAT2. These are a little more permissible for access. You don't need to be behind our firewall to see the data, and they're primarily built for transient usage, either for bringing data from other sites here to MCW to then integrate them into SIR-XNAT1 or SIR-XNAT2, or for sending data out to other studies or other collaborators from other institutions. Additionally, GWXNAT2 is built in such a way that it has some advanced visualization tools, which we'll get into a little bit. The XNATs in general have a hierarchical organization, so you can navigate into a project and then to a subject in that project and then to a particular imaging exam in that for that subject. And on the project level, you have access, management of the user access to it. So only the people that are in XNAT that you give permission to as the project owner will be able to see that data. Of course, as I mentioned above, on campus using SIR-XNAT1 and SIR-XNAT2, we definitely do have great integration with the Research Computing Center here at MCW for processing your data in bulk. Once the data is organized, then the real fun of annotation can begin. So annotation of the data set can actually occur in the GWXNAT2 system. So I have a video screenshot of it here. Once you're in the subject, you can go to the view session, which will open up the OHIF viewer. And on the left of it are going to be the number of series that are in your acquisition. You can drag one of those series into the viewer. And in the viewer, you can scroll through the images and change your window level and contrast as you can in most DICOM viewers. Once it's at a good place, you can go to the Contours button in the top right, click it, and select ROI Contour, and create a name for the new contour that you're about to draw. Once that ROI is created, you can go to the ROI tool, select it, and select Draw. Then, on the image, you can click and drag to draw the region of interest that you care for. As you can see, the precision doesn't need to be high because you can go back and sculpt it by going to the ROI tool again and selecting Sculpt, and then you can push the ROI points by clicking and dragging. With that, you can fine-tune the edges of the ROI. You can both push from the inside and push from the outside. You need to take care to make sure you don't make any loops in it. And by that I mean making something that looks like that, where you have that little notch that's back into it. You can create a closed loop with the Sculpt tool that can ultimately lead to some challenges with your regions of interest. So you need to nudge that back into place. Once you're through with drawing the region of interest on one slice, you can go a few slices over. Again, draw the region of interest on this next slice. And again, you can sculpt the region of interest to make sure that it lines up with those contours that you're aiming to create for your ROI. And then once completed with this, the XNAT ROI drawing tool that we're using here has a nice function that allows you to interpolate the ROI from slice to slice. So here you saw that we drew on one slice, skipped a slice, and drew on the other slice. And now as we scroll through, that intermediate slice has the ROI drawn automatically for us. When done, you can click the Export button, give the ROI a name, click the XNAT button that's there, and then when you go back into the project, it'll appear in the assessment section here. So once we refresh this page, there it is. You can click into it and see that the ROI is defined as an AIM XML file and an RT structure set. The RT struct is used then in our subsequent processing for creating the annotations for the deep learning. With annotation complete, the next step is to identify and train the model. Well, highly efficient and highly effective deep neural networks are a very active region of interest right now for development. Very high-performing algorithms have been released as open source, and NVIDIA has done a great job of bundling these networks up for our use and putting them into a really user-friendly form. They have used the containerization software Docker with their own branch of it. And that container holds all the software dependencies in the model itself and is optimized to use the GPU resources. With that, you can then launch the models on any system where you have this NVIDIA Docker installed without really needing to worry about the dependencies of the software. Specifically here for the segmentation, we're using VNet Medical for TensorFlow. VNet itself is a de facto model architecture shown in the schematic diagram here. It's essentially a three-dimensional UNet, which is, again, the de facto standard of the UNet is for 2D image segmentation. So the workflow for doing this then is once we've annotated that data, we can put that training and test data into a folder that's on our local computer that's shared into the Docker image, build a JSON file that just lists what training data and label data there is, and run the application. The training proceeds without any further interaction from the user. With training complete, the next step then is to test the model. We tested the model by applying it to data sets that were not included in training. Because the outputs of the model are standard NIfTY files, standard neuroimaging viewers like AFNI were easy to use to assess the results. As it can be seen here, segmentation worked very well. If the results of testing are suboptimal, one can iterate by manually resegmenting failed cases and adding them back to the training set in a human-in-the-loop format. Now, in cases where iteration is required, the output of the model can be formatted as nifties. Those nifties then can be edited using something like AFNI's draw dataset. And then, because the model takes nifties in, those modified masks and the underlying anatomy can be put back into that training set, and training can proceed again. Or you can get lucky, as we did here, where our first round of training actually did not require any further modifications of the masks, simply because it worked. So with the deep learning segmentation model finally trained, tested, validated, we can go on to use it. So with the segmentation complete, now we can actually do the science. So as a reminder here, what we're looking at is doing a cross-field and cross-subject registration of instrumented hips. So the first step is to use this deep learning segmentation model to segment out the instrumentation. Then align the centers of mass of that instrumentation to get that first guess. Then define a region of interest where we actually really want this precise region, registration to occur. And then use the tools of simple ITK, which are the underlying registration algorithms that are in the popular neuroimaging registration software ANTS, to do the registration in that region of interest. And then for our cross-field and cross-patient comparison, we put together a workflow where we would align the 3 Tesla image for a patient to the 1.5 Tesla image of that patient, and then align that 1.5 Tesla image of the patient to the template 1.5 Tesla image, and then finally apply that joint transformation of the 3T to the 1.5T, and then from the 1.5T to the template to get that 3T image into the template space. The images on the left here are just examples of the inputs and the process. So on the top left is the 1.5T image of the instrumented hip. On the top right is the 3T image for that same patient, where you can clearly see that alignment of the patient was not the same between those two scanners. The bottom left shows the acetabular region of the implant where the registration was optimized, and the bottom right shows that shifted 3T image in the same space in that acetabulum as the template image. Now, because the segmentation software is actually hot off the press, having finished development on Friday, we're going to switch over to the underlying manually segmented data set here that really motivated our work. So once the segmentations and registrations are completed, one can flip everything over to that left-hand side and create an average image of that template space for the instrumented hip. With all implants then in that template space, we're able to create probability maps of a given voxel having an artifact associated with the implant in that template space. On the top left, you see the general probability map across all implants of an artifact being greater at 3 Tesla versus at 1.5 Tesla. In the top right, we consider metal-on-poly implants, which are a subclass of construction. And you can see that the probability maps still show greater artifact at 3T in comparison to 1.5. In the bottom left, the metal-on-metal implants are shown to have most artifact at 3T in comparison to 1.5T. And in the bottom right, the ceramic implants have the least amount of excess artifact at 3T compared to 1.5T. In that template space, a region of interest around the acetabulum can be defined, and the number of voxels that are artifacts can be counted in those regions of interest. Then you can compare the artifact volume at 3T to 1.5T across the different types of implants. With the metal-on-metal implants, there is significantly more artifact at 3T, and there's a trend to more artifact in the metal-on-metal, metal-on-poly. The ceramic implants show almost no difference between the two field strengths. So in summary, deep learning is becoming ubiquitous in medical imaging, and the technologies have matured to the point where one can either do research on the technology, it's still ripe for more development, or do research using the technology, do research with the technology. And our initial experience of doing research with the technology really illustrated a high barrier to entry in using these segmentation algorithms. So in the Center for Imaging Research, we've developed a more user-friendly workflow for curating data and training deep learning-based segmentation models. And we have infrastructure here with the help of Radiology for data extraction from PACS, And then in the center, infrastructure for image de-identification, data organization through XNAT, image annotation using our GW-XNAT2 viewer, network programming using those existing NVIDIA Docker solutions, network training in those Docker containers, and then model evaluation, viewing those images as they come out, being able to look at those nifties, and in fact edit those nifties and add them back to the training set to iterate on that, retrain the model until you get to the point where you're ready for model application. Using that workflow then, we trained somewhat of a proof of concept model here to segment the hip arthroplasty and use that segmentation then for the registration of implants. And with that study, we showed that implant construction matters when triaging patients to 3T versus 1.5T systems. Finally, I'd just like to thank the team who's responsible for this technology, this infrastructure, and the science that we're doing. So the programmers, Brad and Robin, the ones who have made XNAT and actually put all of those pipelines from crazy ideas that the PhD folks have had into actual robust implementations. We couldn't be doing it without them. The study coordinators, the people who have been really driving and getting the participants into the scanners for us and managing the regulatory side of things for those somewhat complex IRBs that we have to pull the data from the clinical record. Christy, Melissa, Rachel, and Tina. For the CIR team in general, Debbie for making sure everything runs, Jody as well, making sure everything runs, Matt taking care of a lot of the XNAT stuff for everybody that's here, and Kathleen for pushing things forward with the preclinical system. The CIR techs, all of those 3T images were expertly acquired by them. So again, you can have the greatest software in the world, but if you don't have good data acquired by good people, you are going to be lost. The current summer students, Claire and Skylar, are both working on this segmentation net. Claire creating some things for further augmenting the data, make the training even better than what it is right now, and Skylar for really probing into the VNet architecture to see what is good or right set of hyperparameters for the work that we're doing. The radiology team, Diana Kane, she's the one who is able to help us get those accession numbers for pulling the images, and Peter Johnson, who's the very dedicated student here who's drawn the ROIs for those hips using that web interface that we saw. Because we're leveraging the Research Computing Center, it would be remiss not to mention them. So Bill Branson and Matt Flister, who are system administrators there, Matt's the manager there, and David Hotchkiss, whose vision here as the CIO really put research computing on the front burner and led to all sorts of amazing hardware being available for us to use, including a large number of GPUs. Of course, the MCW faculty, for the work that we've done here, have bounced a lot of ideas past Kevin and Tugan. And of course, all. There's a bunch of faculty here. If I list everybody, I fill up a few pages. But we all have some great scientific conversations, and I'm hoping that we can have those over lunch and perhaps drinks at some point here as the world opens up again. And everyone who's listening to this here, thanks for the support.